एलिजाबेथ की है ये नहीं क्या कनेक्शन करिए मेरे काम में दबा एक तो कुछ गेंजी नहीं बनना
नमस्कार। Actually, my laptop is not working. That camera is not working, so I have joined in my phone. So after starting of this presentation, I will switch off this. I will off this and I will talk from my laptop. I think
madam, madam,
namaskar. Namaskar, namaskar. Actually, I have joined it from two devices. That's why echo is there.
Principal sir, do you hear me, sir? Principal sir. Principal sir, do you hear me, sir?
sir join करना थी।
ओहो, अच्छा
हेलो, join करना थी।
ठीक है।
नमस्कार।
हाँ नमस्कार प्रभात। अच्छा भल अच्छु तो हाँ नमस्कार हाँ ठीक है ओ धन्यवाद
गुड मॉर्निंग।
गुड
गुड मॉर्निंग प्रभात। आ कौन के अच्छा कुआ
हाँ भलो
हमें केमती अच्छा
मैडम कौन है
बहुत आगे रहता भलो रहता
बहुत आगे नुह बहुत पछे जाइयो
ନାଇଁ ନାଇଁ ବହୁତ ଆଗରେ ଅଛ ସବୁଦିନ ମୁଁ ଦେଖୁଛି ଅନ୍ୟ ସାରଙ୍କର ସବୁଦିନ
କଣ ଏମିତି ସମସ୍ତଙ୍କର ଠିକ ଠାକ ଅଛି ଆପଣ ଯେମିତି ଇଏ ଆସିଛନ୍ତି
କରନ୍ତୁ ଅଲ୍ ଦି ବେଷ୍ଟ
हाँ
ଠିକ ଠାକ କରୁନାହାନ୍ତି
ठीक है
To all of you, Sakil Babu, should we start now?
Yes sir
Hello?
Sir do you hear me sir do you hear me sir
Yes.
sir
You are
sir
nicely audible.
ठीक है sir thank you sir sir good morning to all of you I Mohammad Shakil IQAC coordinator of Subhadra Mahata Mahavidyalaya first of all welcome you all to the today's webinar whose topic is medicinal plants and modern approaches towards preparation of effective Ayurvedic formulations. At the outset, I would like to express my sincere gratitude and welcome our principal, Dr. Dilip Kumar Misro sir, who has taken all initiatives in conducting this webinar. Sir has contributed a lot,
ଆଜ୍ଞା ସାର୍
and his initiation actually helps us to move ahead. Again, I welcome today's chief speaker and eminent resource person, Dr. Sagarika Parida, who is the Associate Professor, School of Applied Sciences, Centurion University of Technology and Management.
ଆଜ୍ଞା ସାର୍ ଜାଣିଲି
So, ma'am,
ହଁ ସାର୍ ମ୍ୟୁଟ୍
just is teaching to undergraduate and postgraduate students
ଲାଇଭ୍ ଚାଲିଛି
for more than 15 years, including Regional Institute of Education and Kalinga Institute of Social Sciences, Bhubaneswar. So, again, I welcome the Miss Svetalina, Saswati Svetalina Madam, convener, and Mrs. Itisri Samal, co-convener of this today's webinar. I also welcome Professor Padaravinda Biswal, member coordinator IQAC, Dr. Mrs. Suprabha Parida, coordinator IQAC, member coordinator, Professor Biranchi Narayan Ponda, also member coordinator IQAC, and Professor Prafulla Kumar Mahapatra, member coordinator IQAC, who have taken lots of initiative regarding the conduct of this webinar. I also welcome my fellow colleagues of this institution, all staff members of this institution. I welcome professors, lecturers, sirs and madams of different higher educational institutions as well as different universities. I also welcome researchers, professors, educationists and dignitaries across the state. And also I welcome the students of our institution and all other participants of different colleges and universities. So now I request our beloved principal Dr. Dilip Kumar Mishra sir to preside over the meeting. Principal sir.
Thank you, Professor Mohammed Sakil.
Thank you, sir.
Coordinator, Internal Quality Assurance Cell of Subhadra Mahata Mahavidyalaya, and all coordinators, members of IQAC. I welcome you all the participants, my friends from teaching fraternity across the state, students, scholars, researchers, faculty members, all over the state, my colleagues from this college, from different departments. I welcome our esteemed resource person of today's webinar, Professor Dr. Sagarika Parida Madam, who is from Centurion University. I welcome the convener and co-conveners, Professor Ms. K.P. Saswati Svetalina from the Department of Botany, and Itisri Samal. She is also faculty member from the Department of Botany. I again feel proud and privileged to welcome you all to this today's August virtual intellectual activity intellectual program, that is the webinar. As requested by Professor Mohammed Sakil, I am supposed to speak a few lines on this topic, on the relevance of this topic. Friends, let me take back to you to an episode that belongs to, that pertains to Tritiya Yuga, and how Hanumanji had been to Gandhamardan to search for the Vishalyakarani plant, medicinal plant, for curing the wound mortal wound of Lakshman. He managed to come back with the Visalyagrani and Lakshman got cured. That is the, you can say, mythological story we have listened to several times. And it is having more relevance in today's context. Today's topic is medicinal plant and modern approaches towards preparation of effective Ayurvedic formulation. Friends, the more scientifically we advance, the more closure we are getting to the ancient practices. And
सेटिंग देखो ना सेटिंग देखो
I think there is network issues with sir.
Yes, madam. Yes, madam. Some network issue is there. Yeah.
Yes.
Historical background, history of medicine and plants dates back to remote past when herbal treatment was the only answer to all kind of ailments. Nowadays, greater emphasis again being led to phytotherapy. Besides cultivation, setting of herbal gardens are also mooted on hills and plain areas as management of all kinds of diseases is possible through plant drugs without, you can say, sans toxicity. Charaka, the ancient Indian physician, was asked by his teacher to bring plants which were absolutely useless. He returned empty-handed. There was no such plant. Phytotherapy, a treatment with plants and herbs, was considered efficacious than the use of individual compounds. The view is widely held, however, that when the other compounds are blended in with the main active principle, they do, in fact, play an active role through cytolytic action, either by stimulating or inhibiting it. The reason for using the whole plant is that it is impossible to reproduce synthetically, as well as the fact, unproved but suggested by experiments, that is gentle curative action may be due to the low concentration of compounds in it. But this theory lacks scientific backgrounds and doesn't have clear mechanism of action and thus not welcomed all over the world, but as being one of the choice of treatment in Asian Peninsula, and also this region is very rich in these plants. Herbal medicine has still been practiced here from the time immemorial. Nowadays, greater emphasis is being laid to receive it all over the world, and in this respect, a call has been given by the World Health Organization for its revival. The science of medicine and botany have been closely linked from the earliest times, and the contribution of plants in medicines or a history of medicine and plants dates back to remote past when herbal treatment was the only answer to all kinds of ailments. In India, the first information regarding the use of medicinal plants is mentioned in Rig Veda, written between say 4500 and 1600 BC. Perhaps the oldest repository of human knowledge as far as history of medicine in India is concerned. Other ancient books on herbalism are Sushruta Sangita and Charaka Sangita, which give remarkable description of Indian materia medica. However, not much work has been done in this area till early 20th century. The British started systematic identification and survey of Indian medicinal plants in the year 1920. After independence, the Indian Council of Agricultural Research and Council of Scientific and Industrial Research took up all India survey of medicinal and poisonous plants, but this yet is to be fully completed. Again, Publication and Information Directorate, Division of CSIR, has been nominated by UNESCO as the nodal agency for collecting information on medicinal and aromatic plant of the country, considering India has the largest variety of medicinal plants due to its vastness and varying geographic and climatic conditions. Of nearly 2,000 medicines available in the country, only about 200 are of mineral and animal origin, and the rest based on plants. There are 20,000 medicinal plants that can be used for curative purposes according to World Health Organization. And as you know, India is leading member of the Asia and Pacific Information Network on medicinal and aromatic plants. Of late, the South Asia Association for Regional Cooperation, that is SARC countries, emphasized the need for preparing an exhaustive inventory of all medicinal plants in the region, especially these belonging to the forest species. This was decided in the SARC seminar on traditional medicines. The cooperation in the region can be facilitated by an exchange of professional teaching and research staff, improvement of drug manufacturing techniques, and capacities and linkages between allopathic medicinal practitioners and traditional medicinal systems. Traditional medicines or folk medicines are prolific sources of useful drugs, and therefore, great emphasis has now been laid to revive the study of medicinal plants, traditional system of medicine, indigenous drug, etc. Government has formulated the diverse schemes to preserve Indian medicines, to produce herbal drugs, to open a central cell on medicinal and aromatic plants. Large-scale production of medicinal plants are also mooted on Indian hills and plains following scientific extraction of chemicals from them. India grows 1,500 to 2,000 species of medicinal plants in forests and wastelands, most of which have been identified, can be exploited on commercial purposes. Our resource person, Madam Sagarika Parida, is here. She will discuss in length, vividly on this topic, how medicinal plant and how Ayurvedic treatment has become quite effective for treatment of human diseases and all that. So I won't waste our valuable time because the time limit is we can say confined to 1 hour and just 30 minutes or like that. Our participants and they will be participating in the question answer session, interactive session. Now I will request our esteemed resource person to throw light on how modern approaches towards preparation of effective Ayurvedic formulation are on the way. Over to Professor Sagarika Parida, Madam. Sagarika Parida, Madam.
Yes, sir.
Yes. Now you can present your talk.
Yes, sir. Good morning.
Thank you.
A very good morning to you, madam.
Just a minute. Let me share my screen. Then I will start. Hello.
Yes, you are audible. Fine, audible.
Yes.
Okay. Good morning, everyone present here. Welcome you all. Already my introduction has been given by Svetalina madam. I
Um,
think
Madam Parida Madam,
yes sir.
hello.
Hello.
Parida Madam, I'm sorry, let me intervene. I'm sorry for the for skipping another say introductory remark by Ms. K.P. Saswati Svetalina, our coordinator and convener of the program, Ms. K.P. Saswati Svetlana, we would like to introduce our resource person. She will speak few lines about our madam, resource person, madam. Over to Ms. K.P. Saswati.
Thank you, sir. Good morning to all. I would like to introduce the resource person, Dr. Sagarika Parida. Madam, again welcome to our session. She is actively involved in the teaching to undergraduate students and also postgraduate students for more than 15 years, including the Institute NRRI and KISS Institute and also UAT. His research area include mainly isolation of microorganisms, antimicrobial activity, phytopathology, crop growth and production, anatomical, cytotaxonomical study, and powder microscopy for isolation, identification of diagnostic marker cell of herbal drugs. Besides this, she also standardized the herbal drug ingredient used in the Ayurvedic formulation for preparation of monographs for single and compound formulation and indigenous knowledge on herbal drugs. Contributed for 14 monographs published in the Ayurvedic Pharmacopoeia of India. Besides this, she also attend 30 conference and seminar and participated more than 10 workshops and also contributed video classes to the lecture portal developed by DDC Utkal University. Thank you, madam. Again, welcome you to the session. Over to principal, sir.
Yes, now our convener, Madam Saraswati Shetalina, has already given a brief introduction of the illustrious career, academic career of our research portion. Thank you, Shetalina, for your brief introduction. Now over to our esteemed research person, Madam Sagarika Parida. Over to Madam Parida.
Thank you sir. Thank you madam. Good morning
Thank you.
everyone present here. Welcome you all. I think most of the participants are students and already my introduction has been given by Saraswati Svetalina madam. Before that, I start my topic. Now I want to share the PPT. Just a minute. Is this visible?
No, ma'am, it's not visible. Ah, yes, yes, yes. Yeah, ma'am, it's visible, visible.
Visible. Okay.
Yeah.
Now full screen is visible.
Yeah, ma'am. Yeah, ma'am, full screen is visible.
Okay. So today I am going to discuss about the medicinal plants and modern approaches towards preparation of effective Ayurvedic formulations. Before this, I express
Ma'am,
my
please
heartfelt.
excuse me, please.
Yes, sir.
Sir, please enlarge your screen, please.
Screen. Okay.
Yeah. Enlarge your screen. The PPT screen should be enlarged. There is a microphone.
Sir, now it is visible.
Okay, it's all right. Okay.
So before that, I express my heartfelt thanks to Saraswati Svetalina Madam, HOD Botany, Professor Muhammad Sakil Sir, Coordinator at Medicinal, and Principal Dr. Dilip Kumar Behra Sir, Subhadra Mahatab Mahavidyalaya for choosing me as the resource person. Now I am coming to the topic. I will begin from the medicinal plants and highlighting some important points about the Ayurvedic medicines preparation and for its maximum therapeutic effects. So this will be a very brief presentation. Actually, Principal Sir has elaborated about the traditional system of medicines in a very brief and interesting way. And I think it will take more than 1 hour, 1.5 hours. Please, either you can save all the questions till the end of the talk, or if you have any questions, please feel free to interrupt within the presentation. Okay. So let me come to the topic. So we know that as principals are suggested or they said that Ayurveda has been originated in India perhaps more than as much as 5,000 years ago. What do you mean by Ayurveda? Ayu means in Sanskrit its meaning is science. So Veda means science or knowledge. So it is science of life. Ayurveda is science of life. Then before this you should know what are the medicinal plants and does the plant contains all the necessary compounds that will be required for curing some diseases or not. Before that here students are there and I have prepared the PPT for easy understanding of the students also, and based on the habit and habitat, plants are classified as herbs, trees, shrubs, then xerophyte, creepers, epiphytes and climbers. So, so based on the plant types, plants are of different kinds, and based on the life cycle, plants are of also three types. That is annual, biennial. Annual means the plant which complete their entire life cycle within a single season. And then biennial, what does it mean? Biennial means the plant will grow, the vegetative part will grow in a particular season and the reproductive structures will come in another season. So it takes 2 seasons to complete the life cycle. So it is biennial and perennial. Perennial is the plants they can survive for a number of years, and each season the fruiting, the flowering and fruiting will come according to the species. So all of you know that plant kingdom is classified into cryptogams and phanerogams. And cryptogams, they are non-flowering plants, that is Thallophyta, Bryophyta, Pteridophyta, and phanerogams also called as Spermatophyta. That means they can produce seed. Spermatophyta means the plants, they have the reproductive parts or the seeds. So it is again divided into gymnosperms and angiosperms, all of you know, and angiosperms again monocotyledon, dicotyledons, and cryptogams, they are divided into Thallophyta, Bryophyta and Pteridophyta. They are non-flowering. And Thallophyta is again divided into algae, fungi and bacteria. So come to medicinal plants. Medicinal plants also called the medicinal herbs, and it have been discovered, the medicinal plants have been discovered and used in traditional medicinal practices since prehistoric time. Actually the uh, Ayurveda is an Upaveda of Atharva Veda, and sometimes the Ayurveda is also by some traditional schools It is also known as the Panchama Veda. There are four Vedas. Vedas means the science of life. So, Ayurveda is the Upaveda of Atharva Veda, and some also considered this Ayurveda as Upanga of Atharva Veda. Upanga. That means one part, one organ of that Atharva Veda. So it is considered as one Upanga of Atharva Veda in traditional system of medicine in that is accepted by some schools. They consider Ayurveda as Upanga Veda, and it depends upon the body's constitution, that is Prakruti. That means Prakriti of the body and the life forces. What are the life forces? That is different dosas, vata, pitta, kapha. So these are the preliminary basis of Ayurvedic medicine. That means if vata, pitta and kapha, they are in a balanced proportion, then we cannot catch any diseases. So why medicinal plants are important, and uh, do all the medicinal plants contain the compounds or contains the phytochemicals that is required for curing the diseases or not? Yes, all the plants have medicinal values, even if the poisonous plants also have the medicinal values. Okay, how the poisonous plants will be utilized? It is utilized in Ayurveda by purification or sodhana. After purification, the required plant parts are used to make the medicines. So you see, about 80% of the people in developing countries, they depend upon these traditional systems of medicine. And most of the traditional medicines, they are derived from plants and some from animals and other mineral sources also. So this Ayurveda today, it remains as valuable for healthcare system in Eastern world also, because the negative impact of the synthetic medicine or chemical compounds which is synthesized within the lab for curing the ailments or the diseases. So Ayurveda is the science of life, not only in the traditional healing system in India, but now it is accepted all over the countries. And in many countries, there is little regulation of traditional medicine, but the World Health Organization coordinates and maintain the safe and rational uses of these traditional medicine or herbal medicines. So, before going to the preparation of the Ayurvedic medicine, I will show you some important medicinal plants. You see, this is Phyllanthus neururi. This is Bhuiyola. It is used for liver diseases in traditional system of medicine. And this is Cissus quadrangularis. This is called as Hadavanga. It is used for diabetes, obesity, for osteoporosis and Hadavanga. That means it is used to joint the broken bones, and also it is used to overcome the weak bones, to overcome the diseases. That is, calcium is not bounded to the bone, and it has some principle that will initiate. If you suppose you take calcium medicine, you will find that the calcium in urination, it will go out from the body, because some of the active principle is required to bind this calcium into the bone. Bone is a living tissue. So these plants, they have that active principle, and they will initiate to build up the bone in treating the osteoporosis. And this is Embolica officinalis, that is Ola, and it is also used in liver diseases. Then Terminalia bellerica bahada, this is also used to protect the liver and also to treat the respiratory diseases or cough and lower respiratory tract infection and sore throat also. And this terminalia cebula, this is Horida. So Ala, Bahada and Horida, both these three are known as Triphala. And you see the health benefits of Triphala. Triphala is a well-known medicine. It lowers cholesterol and also it relieves from constipation, reduces inflammation, and it boosts immunity. That means your immunity power will be more, and it prevents cancer. Why it prevents cancer? Because amla is rich in vitamin C. So vitamin C is antioxidant. So it prevents cancer, and as amla contains vitamin C, it rejuvenates the skin also. And vitamin C is also required for hair growth and treats dandruff. And also it is known to prevent the viral infection. You see, now in this COVID time, doctors they prescribe generally vitamin C to boost our immunity system. If you take 1 or 2 amla per day, then your immunity will be stronger. It will be boosted. So then come to the next topic. Now I will focus on the Ayurveda. Ayurveda and traditional system of medicine. So why modern approaches, modern standardization processes are required? Because previously ancient age, the traditional healers or the Kabiraja, they used to collect the plants or the plant parts in a particular season, in a particular time, to get maximum efficiency of that particular drug. Either some plant parts like roots or leaves or flowers, they collect at a particular time and particular season. And some of the plants they are collected from in the very early in the morning, because before sunrise, because some of the compounds they will degrade, they will react, or the reaction will occur, and they will convert from one sub compounds to another compound. You might have noticed that if you take that is drumstick leafy vegetables, I mean sajana saga. Am I audible?
Yes,
Am
ma'am.
I
Yes,
audible?
ma'am.
Okay.
Yes, ma'am.
Okay. If we take that drumstick leafy vegetables, that sajana saga, before sunrise, then its taste will be good. It will you feel the that will be sweet. And if you take that leafy vegetable, drumstick vegetable, in the daytime, that is 12 p.m. or 10 p.m., when the sun rays are more, then the taste will not so good. You say that the saga ai gunia laguchi. Generally, mothers they are telling that saga pitkuliya laguchi or aigunia laguchi. What does it mean? That means we know that the TCA cycle, the glycolysis and the TCA cycle. So in night, the carbohydrate converted into starch. So its taste is good. If we collect the leafy vegetables before sunrise, in 12 o'clock photosynthesis is maximum, so other byproducts like alpha ketoglutarate acid, C-saccharidic acid, the byproducts, the medium products in the TCA cycle, they will be more in that leaf, and the taste will be changed. So for collecting the particular plant or plant parts, a special season or a special time is required according to the verses in the Ayurveda or in the that is Atharva Veda. So we know that Ayurveda seeks to normalize body functions with varied techniques, including advice on food and activity. We have to take different foods. If we consume some Ayurvedic medicine, what to take and what not to take will be suggested by the Ayurvedic doctors or the Kapiraja. And it is also involve two processes. One is purification treatment, that is panchakarma, and another is the surgical methods, that is Shalya Chikitsa. So you see the convergence of TSM. TSM is traditional system of medicines. So you see convergence of the traditional system of medicines. These all Ayurveda, naturopathy and Unani, homeopathy, Siddha and Yoga. They converge to a single point. That means all are parallelly, they are parallelly emerges, and each one of these traditional system is equally important and plays an important role for curing the elements and for the betterment of the life. So, so this traditional system of India, the knowledge of history and basic doctrine principle, and now it is globally accepted, and different convergence of traditional system of medicine, they are they are globally now accepted, because they do not have all the traditional system of medicine. If it is properly prepared, if it is properly tested, then it has no negative or side effects. So after this come to three basic principles called dosas. So Vata, Pitta and Kapha. These are derived from five elements of Indian philosophy. So you should know what are the five elements. So five great elements are Prithvi, then Jala, then Agni, Vayu and Akas. You see, Akas or ether is the prime importance, because all other four elements, they are on this earth. Akash has a, or ether has a tremendous force, tremendous energy to uh, fix all these water, air and earth into a particular place on the surface of the water, or some distance of the earth air is present. So this the credit goes to ether, not to the earth, because it has some force that everything have been kept in proper places. So Ayurveda dosas can be identified as regulatory control factors, and these dosas are generated from the fundamental physiologic process in living systems. You see, Vata and its subdosas, they regulate the output of motion. That means regulating input output process. That means input is ingesting some food. You are taking, you are eating. So that is input. Then output, that is feces, urination. So Vata and subdosa regulates this digestive processes. And Pitta and its subdosas regulate throughout or turnover and therefore energy. That means Pitta, that is liver, bile juice. That bile juice is required for digesting the food inside the stomach. And so therefore this Pitta will generate the energy, and this energy will be useful for us to speak, to walk, to run and to think also. And Kapha and its subdosas regulating storage, structure and lubrication. So Kapha and its subdosas, it comes to water element. So that fat storage or protein storage in the blood or sugar storage, that is due to these imbalances in the kapha dosas. So what then, let me come to the modern approaches. Why standardization of herbal drugs are required? You see, it is required to confirm the identity. In Ayurvedic medicine, the plant parts like root, whole plant, sometimes bark, fruits or flowers is required for making a formulation or for making a tablet. So, it needs the confirmation. Is it the bark is from the real plant or not? So it is required. And determination of purity, whether the plant is the right plant or like that, some vernacular names are there, I will give one example that sajana, here it is known as drumstick is known as sajana, but in that is sorry, another example I am giving, that is jammu. Jammu in India, in eastern region of Odisha, we jammu means jammu kodi, that is Syzygium cuminum. But in Berhampur, that is Cydium guajava, that means guava, pijuli. Pijuli, they call it as jammu. So their regional differentiation in the language also is a problem to collect the particular medicine or particular herbal plants or herbal drugs or particular parts of the medicine. So determination of purity and establishing its authenticity. Authenticity means whether the plant is authenticate, that is the required plant or not, and quality and efficacy of the ingredients as well as the Ayurvedic formulations. So for preparation of one Ayurvedic formulation, all the ingredients used in that particular formulation should be qualitatively evaluated, and after preparation of the Ayurvedic medicines or Ayurvedic formulations, that should also be properly that is standardized to see whether all the ingredients are there with proper proportion to overcome the particular disease. Okay, so why Ayurvedic medicine? Because you see, there are some of the elements, some of the diseases are there where no that is allopathic preparations are there. You see leaf 52, even leaf 52 is prescribed by allopathic doctors. There is no liver medicine in allopathic, and allopathic doctors they generally prefer to write leaf 52. No medicines are there in for liver diseases, and also like that septilin. This septilin is an anti-allergic drugs, anti-allergy. If you suffer from cold, continuous cold, then also doctor prescribe this septilin, and there is no such medicine besides the steroids, which is not good at all for your health. And besides steroid, there is no medicine for continuous allergy except this septilin, which is the allopathic Ayurvedic formulation. So regimes of adulteration of drugs, why the drugs, Ayurvedic drugs are adulterated, and what is the benefit of standardization of the drug. You see, all are generally now focusing on herbal products, even cosmetics you see. Herbal, that is aloe vera product, then chandan, and then turmeric. So all are now, they are in a way to jump from the that synthetic medicines to the Ayurvedic or the herbal production. So there is a demand of raw materials. You see, there is lots of that apartments. There is no space for the cultivating land is also is reducing day by day. So there, and all the people they want to be in a safe condition, to want to be very look very beautiful, so they move from the synthetic to herbal drugs. So for that demand of raw materials are more. So there is a chance of adulteration when there is demand and the plant material is not available. So there is a adulteration, there is a chance of adulteration, and this adulteration occurs generally by three interests, either by vested interest, that means they will adulterate and to earn some money, vested interest, or ignorance or lack of knowledge. Like the collectors, the plant collectors, the laborers engaged in collecting the plants, they actually they do not have any idea which plants, the botanical name, or they generally know the vernacular or the regional language, and their ignorance also causes this adulteration. And then unavailability of plants, that is rapid urbanization, as I have already told you. So these plants, day by day, the plants are not available due to rapid urbanization, the multi-storied building occupying the lands or the forest lands and for apartment purposes. So what are the, then these are the regions of adulteration. Then come to benefits of standardization of drugs. What are the benefits? You see, if we standardize the drug, that means the drug is has all the ingredients, authentic ingredients, and has all the phytochemicals to to cure a particular disease, then it can be exported. You see, DXN medicine, DXN is from China. That is this DXN ganosylium, that is derived from one lichen, that is Ganoderma lucidum. And in India, almost every persons before 10 years or 7 to 8 years before, they have purchased this DXN ganosylium, and its cost is 6,055. And in China, this mushroom is known as linji or reishi, and they can export because it is standardized. Otherwise, one, you cannot take one plant from India to any foreign countries with you, because it requires the quarantine taste. So it should be standardized for exporting. Just a minute. Yes. Then standardization and approval. Just in 2020, June 23, you see, published on June 23, 2020, Ayush Ministry stops Patanjali from selling, advertising the purported COVID medicine Coronil. Why? Because the Ayurvedic medicine should be standardized and should be accepted by the Ayush Ministry. That Ayush Ministry is Ayurveda, Unani, Siddha and Homeopathy. This is Ministry of Health and Family Welfare, Ministry of Ayush. So Patanjali, they tried to sell this product Coronil in 2020, and Ayush banned this because it is claimed made by Patanjali of the efficacy of its anti-COVID-19 medicine Coronil were not known to the Ministry of Ayush. So you cannot formulate one Ayurvedic medicine without the knowledge of Ministry of Ayush or Ayurveda, Unani, Siddha and Homeopathy, and you cannot sell. It comes under the Drugs and Cosmetic Act 1940, Chapter 4A. If it is standardized, then it will be accepted and sold. You see, generally in allopathic medicines, what generally they do, if some synthetic medicines will be suppose discovered or formulated by some drug industries, then generally India focuses on the Gulf countries. They will give those medicines to the private hospitals to see the efficacy. Suppose Reddy's lab prepared one, just I am giving one example because I know this already happened. Suppose Reddy's lab formulated one eye drop, and they will target some of the private hospitals and to see whether this medicine is effective or not. They will give the patient their quality control officer will come with certain samples, and they will give freely they will give these sample medicines to the hospital staffs to put the medicines on the eyes, whether to see the medicine is effective or not. If out of 10, 8 shows good results, then the medicine is 80% effective. Then they will again report to their company, and they will again modify the composition to see to make it above 90%. So this happens. So for that reason, always try to purchase the medicine which is available in the market and which is already that is prescribed or standardized and prescribed by WHO, World Health Organization. So, so you see here, this is and another cause of adulteration is by of same name. You see, this is Bacopa munieri. This is also called as Jacoba monieri, this is Bringaraj, and also Centella asiatica, it is also some places it is also called as Bringaraj. But this actual Bacopa monieri is Bringaraj in South India, that here it is called as Thalkuri, but Centella asiatica in South India it is known as Bringaraj. So there is a chance of adulteration. So you see Brahmi and Bringaras. So this is Centella asiatica, Thalkudi, and this is Bacopa munieri. Both are known as Brahmi. And you see here Eclipta alba and Videlia calendulacea. Both are known as Bringaras. Bringaras or Kesaras. But actually Eclipta alba is used and it has some principle for that alopecia. Still Eclipta alba has more secondary metabolites in comparison to Videlia calendulacea, and this can be a substitutive drug, not the original drug. If Eclipta alba, in absence of Eclipta alba, if it is necessary, then we can add, but after quantification of the required phytochemicals, we can add Videlia calendulacea. So, what are the modern parameters of drug standardization? Modern parameters of drug standardization include these points. One is organoleptic evaluation. What is organoleptic evaluation? Organoleptic means it stands for taste, for its color, odor. These are, that is morphological or phenotypical evaluation. Taste. Suppose you will find Andrographis paniculata or chireita. It is bitter in taste. So you can taste it. You can see the plant Andrographis paniculata or chireita. It is morphologically similar or not. You can evaluate from morphological point of view. But if some barks are used, suppose Arjuna chali, bark of Arjuna plant, Terminalia Arjuna, then how could you able to distinguish from other bark? It might have been collected from other plants also. So that is how we will standardize the drug using the modern techniques. So, another is microscopic. When organoleptic evaluation cannot be done, suppose in case of bark, you cannot do the microscopic evaluation, you cannot do the organoleptic evaluation except the taste or odor. You cannot say the bark is not from Terminalia cebula or Terminalia arjuna, or it may belong to any other plant bark. So, in that case, you can go for microscopic evaluation. So, this microscopic evaluation is possible by two methods. What are these? This is anatomical identification and powder microscopy. So, anatomical identification, I will show some of my publications, anatomical identification and then powder microscopy. So, anatomically, if the part, you can take the anatomical structure or the you can take the thin sections of the plant material, you can observe under the microscope, you can find whether the plant sample is belongs to that particular plant or not. So, microscopic evaluation, then powder microscopy is required for this bark. You have to make the bark into a powder form, then you have to make the, you have to prepare the slides and observe under the microscope, then for searching an unique marker cell in that powder microscope, in that bark, whether it belongs to the particular plant or not. You have to compare. You have to collect the particular plant bark and you make it powder, and then you will find whether this matches the unique cell present in the identified plant material matches with this collected plant material or not. If it matches, then it will be taken for the formulation preparation. It will be considered for the formulation preparation of the formulations. Then physicochemical parameters are also useful, like acid insoluble as, then its organic contents, moisture content, these are also required. And also phytochemical analysis. The TLC and SPTLC study will do to see how many compounds are present and what are the compounds. If some plants is having 3 or 4 compounds, suppose that plant is collected from a area where the geographical conditions and the climatic conditions is not appropriate, then the plants will synthesize some stress hormones or stress substances so that it will not match with the standard HPTLC or TLC marker. So in that case, you can discard the ingredients also. Then particle size. For preparation of that Ayurvedic medicine, the minimum particle size to be maintained, and then the test for heavy metals. Suppose some plants, they contain some heavy metals and toxins. That plant is useful, but the plant grows, the plants, they grow on a soil rich in heavy metals, or they grow on a soil where industrial effluents, heavy metals are disposed. On that soil, the plants grow. Then what will happen? The plants will accumulate the heavy metals, and those ingredients, after testing, if it shows that exceeded value of heavy metals with its optimum value, then the plant materials, by the quality control officer, it will just be thrown away. Then microbial contamination. The plants should not be contaminated by microorganisms, because you see, if you keep some leaf and before drying, before preparing the formulation, if it is contaminated, it cannot be used, because all the useful compositions, all the useful phytochemicals will be exhausted by that microorganisms by which the plant material is contaminated. Okay, then test for aflatoxin. You know that this aflatoxin is cancer causing. So after formulation and before preparation of the formulation, all the ingredients, it should be tested for the presence of aflatoxins. Then pesticide residues. Suppose in some plant, like Baidyanath, then Patanjali and Dabur, they have a large number of cultivated field to cultivate the medicinal plants. So in that case, they generally use some pesticides to save the plants from different pests and insects. In in that case also, you have to test after harvesting, you have to test the pesticide residue. If the pesticidal residues exceeds than the optimum value, then also it will be rejected. Then study for shelf life. After preparation, the shelf life, that means what is its manufacturing date and what is its expiry date. How much time the drug will be safely preserved, that should also be uh, quantified, or that should also be tested before that making the formulation, after making the formulations, and that specification should be mentioned. Sorry. So am I audible?
Yes, ma'am.
Yes, ma'am.
Okay. Okay. Then quality evaluation of Ayurvedic drugs. So I have already discussed this organoleptic evaluation, it color, odor and taste, then microscopic evaluation, anatomical or transverse microscopy enables to detect adulterants in crude drugs. So these also detect the crude drugs, adulterants. And powder microscopy where anatomical or transverse microscopy is not possible. In that case, we can identify the drugs under the fragmentary plant material, because after powder microscopy or after making the powder, the cells remain in a form of tissues, group 5 to 50 cells, they intact. That is the minimum particle size. In a single dot also you can, under the microscope, you can you can able to see 5 to 50 number of cells. And based on that particular cell, you can identify whether the plant material belongs to the particular plant or not. Then chemical evaluation, that is phytochemical evaluation and physicochemical parameters. Phytochemical evaluation, it is quantified by HPLC, TLC, and physicochemical parameters are also tested. I will say why phytochemical evaluation is required. Like I said already that in case of that drumstick leafy vegetable sajana saga kotha kahichi, so in that case also, so if this year in one plants 1000 mango is you will get 1000 mango, it is not, you will not be sure that in coming season same 1000 mango will be you will get. That means it is affected by the very environmental parameters and some of the micro environmental conditions. So like that, if some plants they are collected from a specific space, then this year maybe suppose in gingiberine is present, suppose 5% gingiberine we require for making the that Ayurvedic formulations. If from that place we collect the same gingiber officinal or ada, which is called as in Godia it is ada. So if it is in 2%, then you have to maintain its percentage, otherwise its efficacy will not be that standard value. It will not come to the standard value. Accordingly, we have to, that quality control officer have to study the each quantification data and authentication of identification data have to be studied by the quality control officer in the respected drug manufacturing company. So these are the compound drugs and their therapeutic effects. So Marichadi Batti, it is used for cough, asthma, Prihari Batti. These have already been standardized by us, by me. During my postdoc period, we have prepared 24 monographs. We have prepared 24 monographs directed by the Ministry of Ayush, so that these butties and these carrots will be exported by our country to other country where Indians are staying, and also where other other countries people they also want this type of medicine. Like that DXN from China, our people they are going for this DXN medicine. Like that, our medicine is now sold in different countries because it has already, these medicines have already been standardized. So you see here Marichadi Batti, cough and asthma, Prihari Batti, splenohepatodisease, then Lasunadi Batti, that is gastroenteritis and diarrhea, Chitrakadi Batti, digestive problems. So these are different formulations and these are the its therapeutic uses. So these are some Tharo and Rasa. It is Batari Rasa, it is for joint pain, and Kaphaketu Rasa, it is also cough and cold. So these 24 have already been standardized and 20 and already published in the Ayurvedic Pharmacopoeia of India. So we have worked, I have worked on this monograph already standardized. These 24 medicines now can be exported by India to different countries after the standardization, And the Ayurvedic Pharmacopoeia, it has 24 monographs have been published in Ayurvedic Pharmacopoeia of India, part 2 volume 1 and part 2 volume 3. And it now, what happens, what are the monographs? It is a standard, this pharmacopoeia is a standard reference book, and after publication in this Ayurvedic Pharmacopoeia of India, then all the standard parameters that comes, that mentioned in the Ayurvedic Pharmacopoeia of India, that comes on the Drugs and Cosmetic Act 1940 chapter 4a, and it is an act of the Parliament of India which regulates the import, manufacture and distribution of drugs in India. Once the value is incorporated or included in this Ayurvedic Pharmacopoeia of India in the form of monograph, then all the pharmaceutical industry like Patanjali, then Baidyanath, Dabur and all the Sri Sri, the Tattva, all will come, all have to follow, all the drug industry have to follow the guidelines to prepare these medicines for its maximum efficiency. You see, this work has already been done by me and published. You see, this is Chitraka. In Sanskrit it is known as Chitraka, and in Oriya it is known as Chita. Sweta Chita, Plumbago gelanica, this is Sweta Chita. This is Lila Chita, Plumbago auriculata, Plumbago rosea. It is Rakta Chita. So, same plant with different flowers, but the root of Plumbago gelanica is only used in preparation of Ayurvedic medicine. The root of only Plumbago gelanica is used in the preparation of Ayurvedic medicine. So how you will know whether the roots collected by the untrained laborers belongs to this Plumbago gelanica or auriculata or rosea? Just having a simple microscopic evaluation or anatomical study, you can evaluate, you can identify whether the plant belongs to the Plumbago, the root belongs to Plumbago gelanica either or Plumbago auriculata or Plumbago rosea. You see here, Plumbago auriculata, you see the internal structure. Here you see my cursor, here are the stone cells. Okay, and the stele is very large, cortical cells are very narrow in Plumbago auriculata. So this is also Plumbago auriculata. You see, after taking the anatomical sections, you see the arrangement of cells, and you see here Plumbago gelanica. In Plumbago gelanica, here you see the stone cells are formed in a form of ring around the stellar region, around the phloem. And here you see the stone cells, a group of stone cells are present here and there and distributed diffusely in the cortical region. Okay? And Plumbago gelanica has a lot of, it contains a lot of starch, you see, which is not present in Plumbago auriculata. And you see Plumbago rosea, there is no stone cells. Here patches of stone cells are there, distributed diffusely in the cortical region. Here in auriculata it forms a ring, but in rosea it is not there. If you take the anatomical section, and if you find there is no stone cells present in the cortical region, then you will be confirmed that this is not our destined root, or this is not our root which we want to make the formulations. And this article is published by me in Bioscience and Biotechnology Research Communications in special issues. Okay. Then come to, I have done also this work. This is on Tulasi, that is Ocimum sanctum. Ocimum sanctum is Krishna Tulasi. Okay, and there are other varieties of Tulasi, that is Ocimum canum, that is Kannaka Tulasi, and the white variety of Tulasi, that is Osimum sanctum, this is Radha Tulasi, and this is Krishna Tulasi. So this is Radha Tulasi, then Rama Tulasi. Rama Tulasi is Osimum gratissimum, sorry, Osimum gratissimum, and it has large leaves, but smell is exactly like that Osimum sanctum, and it is Osimum basilicum variety purpurascens, and it is Osimum basilicum, that is Sugandhi Tulasi, and it is Durlabha Tulasi. So these all belongs to Osimum. So I, on the basis of, you see the morphological difference, but if we want the stem to prepare the Ayurvedic drugs, we cannot distinguish morphologically to which one is the correct, that stem belongs to Osimum sanctum or not. So in that case, we can go for morphological identification, sorry, anatomical identification. You see, this is Kannaka Tulasi. Here stone cells are occupied in the four regions, Kannaka Tulasi, then this is Radha Tulasi. You see the their anatomical differentiation, a lot of anatomical differentiation. A number of trichomes are present, and here that the xylem and phloem is developed much more than this. You see a ring of xylem and phloem, but here some region it is confined to only the ridges. Okay, and Osimum sanctum, you see the difference between Osimum sanctum, that is Radha Tulasi or white variety, and the Krishna Tulasi, that is black variety. You see the difference. Here you see the that is xylem and phloem is not much more developed like that Radha Tulasi. Radha Tulasi is, it is generally confined to the ridges, but it touches with each other. But here you find very less, just only it is confined to the ridges only, like a patches. Okay, and Osimum gratissimum has a well developed because it has broad leaved and it is like a shrub in comparison to these three. So large shrub. So this has automatically a wide region of conducting cells and xylem. This is xylem, and here stone cells are, patches of stone cells are present. You see here, a band of stone cell is present, here patches are present. Like this in Durlabha Tulasi also you will find the difference between this and this. It is different, very different in microscopical study, and this is Osimum basilicum, Sugandhi Tulasi, the ridges are and furrows are very deep here. It has also been published in one of the paper, in one of the journal, and then powder microscopy and why, and that is already we have talked about the anatomical features. Then come to powder microscopy. Why and how? Why powder microscopy is required? Already I have suggested, I have told, discussed about the bark. We cannot distinguish the bark. If some bark is used in the Ayurvedic formulation, we cannot distinguish. So we have to make powder like that. If some dried flowers are there, or if some very tiny seeds are there, we cannot distinguish similar kind of cells, similar kinds of seeds from each other by looking its morphological features only. Okay. So you see Apium graveolens, this is known as Sabitri, and Trachyspermum roxburghianum, this is known as Juan. Okay, and both the seeds are morphologically very similar to each other, and this has been published in World Journal of Pharmaceutical Research, already published. I have worked on this. So we can also visualize the microscopic features depending upon its powder microscopy. That seeds also shows the powder microscopy, the results under the microscopical view, both the seeds are variable from each other. So particle size for the powder drug, very fine. If you take very fine particle, it should be less than or equal to 180 micrometer. 180 micrometer. Micromilliliter. So it should passes number 80 sieve. So if after powdering you have to sieve it in 80 sieve, then you will get this amount of this diameter of this powder drums. And according the sieve size should be to get the desired diameter of the powder particles. You have to sieve the powder material in variable number of sieves, and then the whole plant parts and the whole after preparation of the formulations, this should be tested, the tablet should be tested for the types of microorganisms. If the tablet is contaminated with the microorganisms, then it will be totally rejected. The prepared batch should be rejected. So you see the types of microorganisms. Total aerobic microorganisms should not be more than 10 to the power 7 colony forming unit per gram. If you take 1 gram of that particular drug, you can culture them if to show, in order to show the presence of microorganisms, then it should not be 10 to the power 7 per colony forming unit per gram. If it exceeds beyond this 10 to the power 7, then the formulations will be rejected, cannot be sold out. So for yeast and mold, it is 10 to the power 4 colony forming unit for the plant material for use as teas and infusions. We are taking teas, so that tea also, the dried leaves also, they should not have more than 10 to the power 4 colony forming unit per gram of that is of microorganisms in the, suppose in the tea. If you take the tea dust, if we culture it in the culture plates for ensuring the presence of microorganisms, the colony forming unit or the microorganisms should not be exceeded from 10 to the power 4. Because we are taking internally, we are taking every day, we are taking tea. You see, under the microscope, powder microscopic characteristic of cannabis sativa seed. Cannabis sativa seed, it is in Odia it is known as bhango. ଏଇଟାକୁ ଓଡ଼ିଆରେ ଭାଙ୍ଗ କହନ୍ତି. So that seed, how we will evaluate, actually this seed is cannabis seed, it belongs to cannabis sativa or not. So in that case, powder microscopy will be a valuable tool. After making the powders, you will see the fragments of the sclerenchymal layer of the pericarp. You will see like this type of cells. These are the stone cells with very that is with furrow and ridges. The thickening is like this, and in that monograph, or for publishing in the monograph, or for acceptance in the Ayurvedic Pharmacopoeia of India, we have to draw this using the camera lucida microscope in the microtic pen, and this is the drawing drawn using that camera lucida microscope, and this is sent to Ministry of Ayush for its identification and authentication. You see, and we know this is microscopic characteristics of latex. You see how we will that differentiate of some latex, suppose Calotropis procera and Calotropis gigantea. That means dhala arakha and bayon arakha or purple arakha. But Calotropis procera is useful in Ayurvedic preparation. Calotropis procera or dhala arakha is more useful than the purple variety. So then the Calotropis gigantea, that is purple variety. So in that case also we can do, if some differentiation is there, we can also observe the characteristics of the latex. Here this is the name I have not given, it is because it has not been published. It is some Euphorbiaceae family, some latex in one, suppose in Euphorbia hirta. Suppose I have not written here the names because this is the work going on. You see the starch, we know that starch after adding iodine it looks blue or black. You see the starch grain with dumbbell shaped or bone shaped starch grain here. It takes iodine stain. That means it is dumbbell shaped. That means it is starch grain. It is confirmed that these are starch grains. And in other variety, same plant euphorbia like that I have told you, calotropis procera and calotropis gigantea, like that dhala arakha and purple arakha, like that it is of euphorbia, that siju, and it is also the latex of some siju, and it shows raphides, that is calcium oxalate crystals in the form of fine needles. It is raphides, and here you see the tiny sparkling objects. These are rosette crystals in the form of rosette like structures. So these are the modern parameters of evaluation. This is aloe vera, that is leaf juice, aloe vera leaf juice. We have to draw this microtic diagram for the needles of calcium oxalate, and this is the TLC. TLC for presence of aloin. We have to extract this, then we have to run the TLC plate, and we will get the
से बंद कर ले
image. Sorry. So you see it is Guduchi, Tinospora cordifolia, and its moisture content, total IAS, and its unique cell present in the powder microscopy. You see here the cells contain that tetrahedral shape calcium oxalate crystals. Within the cell, calcium oxalate crystals are there, which is rectangular in shape, and also xylem fibers. So you can distinguish from the unique cell structure. Likewise, we are also different from each other. Human beings, you see, human beings, there are majorly four races, that Negroes, that Mongolians, then white Caucasians and black Caucasians. You see, Negroes or South African people, they are very different from us. And we black Caucasians are different from that white Caucasians, Europeans, because there is a mutation in the 19th chromosome in Mongolian peoples. That means South Korea, China, then Nepalese, and in our northeastern region of India also, Mongolians are there. So in the 19th chromosome, there is a mutation leading to white skin color, straight hair. You see, they have very black and straight hairs because they are Himalayan base. Their eyes are not grey, their eyes are black, their hairs are also black, but their skin color is white, and their eyes with overlapping that overlapping eye iPad, overlapping iPad. So they looks that their eyes looks dropping down like that. So this is due to the mutations occur in the 19th chromosome. Like that, you see variation in every living means in every organism variation is there. You just see mangoes, Alfonso, Baiganapalli, and you see rice, Dehradun, Basmati, Lalata, Jaya, Padma. So a number of variations are there in a single species. So we can, all are variable, we can differentiate which one is required for us for making or for preparation of the particular drug. And you see, I have also worked on this Bansalochana. It is Bansalochana is it is known as bamboo manna. Bamboo manna, it is a siliceous material. Siliceous material present in Bambuja originancia. That means in Conta bouncer, which is generally available in the Himalayan region, in the internode, in the empty spaces of that Bambuja originancia, these siliceous materials, they are present inside that, and this siliceous material is used in Ayurvedic formulations in as to relieve from cough.
ଏଇନା କିଛି
itching, dry throat and cough. And this
ଏଇନା କିଛି
siliceous
ହେଇପାରିବ
material
ନାହିଁ ଆଜି
is
ଯଦି ଲାଷ୍ଟ
generally
ଡେଟ୍ ଏଇ ଆସିଯିବ
adulterated by
ଯଦି
the
ଟୋଟାଲ
silica.
କଲେଜ ସବୁ ବନ୍ଦ କରିଦେବ ଠିକ୍ ବନ୍ଦ
So how you will identify it? Looks after grinding, it looks like that silica powder. So we have standardized this siliceous material, this Bansalochana, and the article has already been published in Indian Drugs, that is one SCI journal, how to identify this siliceous material from the synthetic silica. And this is various variable photographs of unique cells of different ingredients in Lasunadi bati. Suppose this is Piper nigrum, that is golmaricha or black pepper. This is gingiber officinal, you will find oleoresin cells, that is gingiber officinal, that is ginger or adda. This is cuminum cyminum jeera or cumin seeds, allium sativum, that is rasuna garlic, and piper longum pepper. So these are the microscopic evaluation of the powder drugs after making the powder, and these are the standardization based on the phytochemical parameters. So these are different, you see T1, T2 and T3 and T4, and T1 is maricha, that is black pepper. We have to write in Ayurvedic Pharmacopoeia of India, we cannot write, we will write its Sanskrit name maricha, and we have to write its scientific name, that is Piper nigrum. Other names, English name is not required in for publishing in Ayurvedic or for to work with the Ayurvedic Pharmacopoeia of India. Then T2 is Lasnadipati, it is Maricha, and it is the overall preparation formulation. And T3 Lasnadipati, it is Vaidyanath. So we are in a process of comparison with the product from the Vaidyanath and from IMMT, which we were preparing that time to standardize the formulations. Then these are different, T4 is Pipali, so Maricha and Pipali, that means Piper longum and Piper nigrum are there, and both the bodies are there, both the tablets are there, and this is the SPTLC fingerprinting. It shows different peaks of different compounds. Like this, it is Marichadipati, and this is also. So here another interesting fact is that now Ayurvedic research is moving forward. It is going on, continuously going on for the benefit of human being. So you see here Marichadipati Vaidyanath is prepared by Vaidyanath, and it is prepared by us, RRL Bhubaneswar. That time it was known as RRL, Regional Research Laboratory, where I did my postdoc, and now it is known as Institute of Minerals and Materials Technology, and it is prepared by RRL Bhubaneswar. And also T3 without jaggery. So Ministry of Ayush has given the task to prepare without jaggery. Without jaggery, so why without jaggery? Because it can be taken by the diabetic people. After preparation and standardization, this can be, so this is based on the preparation is based on the ancient formulas. The verses in the Upanishad, or the verses in the Rig Veda, according to that we have prepared, according to the literature we have prepared, but according to the directions of the Ayush Ministry, we have prepared without jaggery for the benefit of diabetic people. So this is Baschanava, Aconitum jasmanthum. Baschanava, it is in Odia it is known as Bhalia. Bhalia. Bhalia is poisonous. All of you know, in village all of you know about Bhalia. Some interesting stories are also there about Bhalia, but Bhalia is, it is in Sanskrit, it is known as Baschanava, and this Bhalia is poisonous. But what to do? But Bhalia is required in some formulation, then what to do? We have collected, at that time we had collected that urine of hippopotamus from Nandankanan. By paying fees, by paying some amount of money, we generally do collect the urine, and then this Vaschanava or this Bhalya will be kept for consecutive 3 days, 72 hours in the hippopotamus urine. Ganda ପରିସ୍ରାରେ ରଖାଯାଏ for purification, ଶୋଧନ. Then it will be useful for preparation of Ayurvedic medicine. You see here thin layer chromatogram of Marichadipati with and without jaggery. Here with and without jaggery already it has been standardized and already available in the market also. So this is powder microscopy. In Sanjeevani Bhatti a number of plant based ingredients are there, number of plant based. So you have to, after preparation of the formulation, you have to take one bhatti and you have to examine, you have to prepare a number of slides, not in single slide you will get all the unique marker cell of the ingredients used in the Sanjeevani Bhatti. But you have to prepare a number of slides, you have to authenticate that these debris or these type of unique marker cells are present. Then after that the body will be it will be packed and the label will be pasted for marketing. This is also Plihari body. So these are different manufacturing process, how we will prepare the kharo, and this is different manufacturing process of different rasa, Lakshmi Vilasa rasa. These are the Sanskrit name of Datura, means Dudura, Bhango, Cannabis sativa, Satabari, Acorus calamus, Nagavala, that is Sida, Cadifolia, then Atibala, Sida, Cordata. So like this different plant materials are used for preparation of Ayurvedic formulation. This is also another rasa, the manufacturing process of Amlapitantaka rasa, and these monographs have already been published by us, and all the pharmaceutical industry engaged in preparing the pharmaceutical formulations or the Ayurvedic medicine, they should follow these rules. So what are the applications? Last come to the last part of the Ayurvedic medicine. What are the applications? So quality control of Ayurvedic single and compound formulations. After evaluating the single ingredient, we can evaluate its quality for its better efficacy, and publication after this monographs will be published, which will come under Drugs and Cosmetic Act, Chapter 1940, Chapter 4A, and the quality control will enhance the efficacy of the formulations. It will enhance the formulation's efficacy. Suppose you are taking one medicine, definitely it will cure the particular dosas or the metabolic disorders or physiologic disorders. Then enhance the economy by exporting the formulations. After standardization, then we can export this medicine to other countries, and by this a non-resident of India and people from other countries will also get the benefit. Then thank you. This is our university gate. Okay. Any questions you can please ask me. Hello.
Yes, ma'am. Hello.
Hello, sir. Yes, you are audible.
Yes, yes. Hello.
Hello.
Yes, sir.
Yes. So, uh, madam, can you go for the question answer session now?
Yes, yes, sir.
ଆପଣଙ୍କ mute ଅଛି
Okay.
camera.
Uh, we have with us our IQSA member and also one of the organizing committee members, Professor Padaravinda Vishwal. Padaravinda Vishwal, are you here?
Yes, sir.
Hello?
Yes, sir.
Yes.
I'm here.
So you can now conduct the uh, uh, question answer session. Invite questions from queries from our participant friends. You can do that.
Okay,
Over
thank you,
to
sir.
Padaravinda Vishwal.
Thank you, sir. So, thanks, madam, for your nice presentation about the medicinal plants and their effects on our body. So, my dear participants, anything you have in your mind or any question at your end, please the queries can be clarified by our resource person, Dr. Sagarika, ma'am. So, any question, queries from our student side or from any faculty members present over here? So, by the way, ma'am,
Shall I stop the screen? Shall I stop the sharing of the screen?
yeah, you can stop also.
Yeah, ma'am. Yeah, ma'am. Yes, ma'am. Yes.
Okay.
So, ma'am, so I
Yes,
have
sir.
a question in my mind.
Okay.
Since ancient times, so Indians are more exposed to that of the medicinal plants.
Yes.
So, like Chinese believe in this also in some extracts of the fungal or micro or any other algal products, all these things. So, do you have noticed any adverse effects of those medicines or the herbal medicines on another disease or any body parts of our body parts?
Sir, actually,
Those
the
dosage or
standardized
any other medicine specified for any other treatment of the any other disease, and some have some adverse effects.
Sir, if you see, if the particular medicine is properly standardized and available in the market, then it will not cause to initiate or to have a adverse effect. If it is not properly standardized or some adulterations is there and some pesticides are there or some heavy metals are there, definitely they will cause some harmful effect.
Okay, madam. Okay, okay. So,
That's
ma'am,
why, sir,
another question is
nowadays,
there.
yes, sir, yes, sir.
Yeah. Please do answer, please.
Sir,
It
another thing is
depends
that,
upon the dispensation. Yes.
yes, sir, for that reason, quality control officer in every pharmaceutical industry, they have to follow the guidelines according to the
Yes.
pharmacopoeia, according to the Ministry of Health.
Okay, okay.
Good.
So, ma'am, so we are here, now running through a very adverse situation due to the effect of the COVID-19.
Yes, sir.
So, when the product was produced by Patanjali Yoga Pitha
Yes,
Haridwar,
sir.
I think the
Yes, sir. Coronary, sir. Yes, sir.
already you mentioned that,
Ha,
but
ha, ha.
it was truly objected by the so many of the medicinal, so medicine farms producing the allopathic medicine.
Yes,
So,
sir.
is there any ministerial or government level hurdles to market that particular medicine during that corona period?
Ha,
And Baba
sir.
Ramdev was telling that it is very effective for the treatment of that particular COVID-19
Yes, sir.
case.
Yes,
So,
sir.
this, yes, ma'am. So,
Yes,
ma'am,
sir.
my
Sir.
question is that the ingredients contained in those the extracts of those medicines are very much difficult to extract all these things in a raw form. Common medicine are available common medicines are available for the treatment of the cold and cough in our local area, and
Yes.
the Ayush Mantralaya has not duly recommended that medicine for the treatment of corona till now.
Son, no, sir. Initially in 2020, 2020 June, the ICE ministry has banned because it has not been standardized. Because coronil is not mentioned in any Ayurvedic text, and they have prepared on the basis of the experience that on the basis of the plant materials for their properties described in different verses of the Ayurvedic granthas or that Nibandhus. So they have taken those plant materials. Suppose the Nyctanthus arboretritis, or you called it as Cephali, or that is Ganga Siuri. So that is taken for relieving that, that is antipyretic. That means antipyretic means it will reduce the fever. So like that, some of the ingredients are mentioned for its some of to that balance some of the symptoms like cough, like cold, then fever, and strengthening your lower respiratory tract. So what they have done, they have taken all those ingredients from the text, ancient text. They have taken those ingredients and prepared their formulation to boost the immunity, and named it as Coronil 19. So Coronil tablet. So Coronil at that time no evidences were there in Ministry of Ayurvedic regarding its standardization of efficiency. After that, the preparation of medicines, they have tested, standardization has been done, and they have tested in different animal model, and then already it is available in the market, and it is also effective. It is proved as effective. So based on the new medicines are have been prepared based on the ancient text, that individual ingredient, its properties, and they will mix, and new formulations have been produced.
Okay, thank you. So, my dear participants, any other question?
Yes, sir, I have a question.
Thank, thank you, madam. Thank you, madam, for your valuable
Thank
presentation. I have one question,
Yes.
madam, Satrika, madam. I have one
Yes, madam.
question.
Yes, madam.
How can we test the presence of this aflatoxin in the herbal products?
Uh, Yes, madam. That is on that in that formulation, in that industries, they have different types of instruments. They have, after making the powder like that. Suppose one tablet is formed, and this will be tested before releasing. This will be tested for it solubility test. If the tablet after we take, it will not be soluble, dissolve within the stomach within 15 to 30 minutes, then it will the tablet is wasted form. If it is not soluble, then again they will prepare this. So like that, they have different that instruments for a single batch. How many batches will be produced? Every batch will be standardized. Every batch will be that quantified for different pesticides, heavy metals. The ingredients will be quantified, verified, and the tablet will be also be verified for its toxins, heavy metals, and pesticides.
Okay, madam. Thank you.
Any other question?
Thank you, madam, for such a nice presentation. I just want to share my views on the topic. For centuries, indigenous cultures around the world have been used traditional herbal medicine to treat a myriad of maladies. Large number of modern medicines are derived from the plants which are used in Ayurveda and other medicinal systems like Unani, Kabiraj. But still, I feel there needs a promotion of traditional Indian herbal products. What is the hurdles in that?
Yes, madam.
Yes.
No, madam. Now also research is continuing, going on. We think that traditional system is now not working, but no. ICE ministry is taking care of that in developing different medicines and developing different high quality medicines for the betterment of people. You see, till now also no liver product is developed by the allopathic medicines. No liver, only lead 52 they will prescribe if you have jaundice, if you have any liver disorders, they will just, the allopathic the practitioners, they will just write the lead 52 medicine, which is totally Ayurveda and produced by Himalayan Ayurvedic company. So it is, we thought that we have a idea that Ayurveda medicine is now no longer is used. No, that is not correct. In each and every, now also different researches are going on in NBRI, CMAP and by proper supervision of this ICE ministry.
Thank you, ma'am.
No, the fact is clear that the Indian-made Covaxin and Covishield were not being accepted by countries like UK and America like that.
Yes, sir.
But when there was political pressure, okay, that got approved.
Yes.
The thing is that our Ayurvedic products have got to be accepted by standardized laboratories,
Yes.
so that when they will be accredited, or you can say get some sort of international acceptance, these medicines can really they have global recognition.
Yes, sir.
So it is completely political, and it is under the control of international standard laboratories. If they approve, then the business will run smoothly and things will get accepted.
Yes, sir. WHO should
Um,
recommend this. World Health Organization
yes,
should
yes,
recommend this. Yes.
Ma'am?
yes.
Hello? Ma'am?
Yes, sir. Yes, madam.
It is, yes.
Ma'am, Tinospora codifolia can use in cure the COVID-19?
Tinospora cordifolia is guduchi, that is actually antipyretic. Jara bhalo huଏ ସେଥିରେ. So it is used. It is one of the ingredients. Tinospora cordifolia, any, and that Andrographis paniculata also, it also cures malaria, and that Aja directa indica, neem leaf also. So it can. So they have chosen different ingredients for different symptoms that because of that coronavirus. According to the symptoms they have chosen, those symptoms will be cured by some of the ingredients. Those ingredients will be, they will taken and prepare the medicine. They have taken and prepared the medicine. Yes, madam.
Thank you, ma'am. Ma'am, a traditional healer can or Kaviraj use the Ayurvedic ma'am, say phytochemical screening and antimicrobial property study ହଉନଥିଲା, but ଯେ ଆମର ବଡି ପ୍ରତି କିଛି ହାର୍ମଫୁଲ ଥିଲା କି ସେତେବେଳେ
No,
ଯୋଉ
they, at that time pollution was not so much high, and they collected at a particular time, madam. They collected at a particular time according to the literature cited. In which period, suppose some of the latex, they will, you see that the latex of that palm tree, जोटाको कहंती आपनकर ताଳ ଗଛ ଖଜୁରୀ ଗଛର ରସ, that after sunrise it will be fermented. So like that, some of the plant parts they have collected from a particular space, particular place and particular time, and that time no much pollution was there, no heavy metals were thrown to the by industries waste like that. Now it is tested for the efficacy, for the betterment of the human being.
Okay, ma'am. Thank you.
Thanks. Any other question? Perhaps no such queries.
Wouldn't find any other query from any participant.
Right, sir.
Okay, so should we really that ask Itisri madam to offer vote of thanks?
Yes, sir. Yes, sir.
Thank you, sir. I
Itisri,
expect.
over to Itisri madam.
Yes, ma'am. Yes, sir. Yes, sir. I extend my sincere thanks to the resource person, Professor Dr. Sagarika Parida, for her gracious presentation and spending some of her valuable time with us, making the webinar meaningful and informative too. Thank you, ma'am. I would like to thank the Chief Patron, our Principal, Professor Dr. Dilip Kumar Misra, for his encouragement and guidance to organize this webinar. Thank you, sir. I would also thank our IQAC coordinator, HOD in Physics, Professor MD Sakil, for his constant cooperation, and thank our HOD Saswati for her moral support. I am equally thankful to our IQSC member HOD in Zoology Professor Padarbindo Vishwal and HOD in Chemistry Professor Dr. Suprabha Parida for her advice to make this webinar successful. I am very much thankful to all the participants who joined with us to make this webinar successful. I also thank to our technical team who helped to conducting this program undisturbed. Thank you to all.
Thank you, sir. Thank you,
Uh,
madam.
Thank you, madam, for your quite rich and intellectually rich and rewarding presentation. We learned a lot of things which you were completely ignorant of. Um, I thank all my organizing team members, IQSC team members, my colleagues, my students, my friends from teaching fraternity across the stage, and all other delegate friends. Thank you all for making the program a grand success. Now the program stands closed. Thank you all.
Thank you,
Thank
sir.
you.
Thank you.
Thank you.
Welcome, sir.
Thank you.
ତୋ ରିସୋର୍ସ ପର୍ସନର ନାଁ କ'ଣ ମୋତେ କ'ଣ କହିବାକୁ ପଡ଼ିବ ମୋତେ ଗୋଟେ ଚିଠା ଦିଏ ତାଙ୍କ ବିଷୟରେ ତାଙ୍କ ପଠେଇଦେବେ ହାତ କରିଦେବେ ହଁ ତାଙ୍କ ବିଷୟରେ ଚିଠା ଗୋଟେ ଦିଏ ତା ନାଁ ହେଲା ମନୋଜ ନୁହେଁ ସେ ବାଲିପେଟ୍ରୋଲ ୟୁନିଭର୍ସିଟିରେ ପ୍ରଫେସର ତା ପୂର୍ବରୁ କ'ଣ କରୁଥିଲେ ତା ପୂର୍ବରୁ ଯେଉଁ କଳା ୟୁନିଭର୍ସିଟି ଆସୋସିଏଟ୍ ପ୍ରଫେସର ହଁ ସେ ତାଙ୍କର ଟିକେ ଡିଟେଲଟା ମୋତେ ପଠେଇଦେଲେ ମୁଁ ତାଙ୍କୁ କହିବି ଆଉ ତାକୁ ଓଡ଼ିଆରେ କହିବେ ନା ଇଂଲିଶରେ କହିବେ ଓଡ଼ିଆରେ କହିବା ଓଡ଼ିଆରେ ଲିଖିକି କହିଦେଲେ ମୁଁ ତାଙ୍କର ଗୋଟେ ଆଡ୍ରେସଟା କହିଦେବି ଆଉ